I keep mentioning E because when we're going to look at disorders, there is one disorder associated with either mutation or absence of ApoE that contributes to hyperlipidemia. Why? Because it results in incomplete binding to the LDL receptor. So you need this primarily plus that. Right? So just file that away. So again, this part hasn't changed at all. It's exactly the same. It's going to deliver the tag to extrahepatic tissues, slowly shrink in size, and become a VLDL remnant. In this case, it isn't called, of course, a chylomicron, but the VLDL remnant is called an IDL, or intermediate density lipoprotein. All right? Here's where they differ now in fate. The IDL will return to the liver, but will only sort of deliver more tag cargo. So here's where the hepatic lipase becomes even more significant, because it takes IDL, and again, we're talking about liver here, takes the tag out, or hydrolyzes the tag, and the particle evolves, stays outside, into the low-density lipoprotein particle, which now has a completely different charge. Again, it still has ApoB100, it still has ApoE, and now this is mostly cholesterol and cholesterol ester. So this is the major carrier of cholesterol to tissues, and all membranes need some cholesterol. All gonads need cholesterol as a precursor, and of course the adrenal cortex needs cholesterol as a precursor to cortisol and aldosterone and androgens. Okay? So again, it starts off exactly the same. One is dietary, one is mostly endogenous. The early phase is similar, identical. Again, you have to have an active lipase. You have to have ApoC2 activating it, low levels of ApoC3, and you have to have ApoE for the eventual fate of this particle. All right, now, this pathway, both of these pathways, are essentially, most of the time, in the fed state. One of the aspects of the fed state, which hormone is high in the fed state, insulin or glucagon? Insulin. Turns out that lipoprotein lipase is induced by insulin. So a type 1 diabetic who is not well controlled will actually have suppressed mRNA for lipoprotein lipase, and frequently they have hyperlipidemia because this is not induced for. Now, it isn't entirely dependent on insulin. So there's always some lipoprotein lipase, because again, you do have VLDL particles in the fasting state, but it is induced during each meal, so you get more lipoprotein lipase expressed at the cell surface. Okay? Makes sense, actually. I mean, this is the scaffold on which the lipoprotein particle is going to dock and empty its content. All right. So let's talk about this particle now. So we've covered its metabolism, its precursors, both of them, all the apoproteins. Okay. So now comes the part that seems to be more correlated with hyperlipidemia than any other, I should say, harmful hyperlipidemia as opposed to benign hyperlipidemia. And that is the concentration and half-life of this in tissues, in the blood, so to speak. So again, LDL particles are cleared very little by the liver, for the simple reason that the liver doesn't really express the LDL receptor. It expresses an LDL-like receptor. So eventually it is cleared. But the favorite target is literally any cell that expresses the, again, LDL receptor. And this could be anything, and I gave you one example of, for example, the adrenal cortex. The fascicularis, so specifically, actually all layers of the cortex. Because again, this is a necessary precursor. You need cholesterol for cortisol. So one of the aspects, remember, of ACTH stimulating the fasciculata was increase in the number of LDL receptors. Remember, ACTH was the controlling hormone of the anterior pituitary, and one of its mechanisms of action was to increase LDL receptor. If you increase LDL receptor expression at the membrane, you would, by definition, increase cholesterol ester uptake. And once the LDL particle binds to the LDL receptor, the whole complex gets internalized. This is released and degraded so you can release its contents. Usually, the LDL receptor recycles back to the membrane to be reutilized in subsequent cycles. All right? And remember, so this was one of the aspects of increased cholesterol uptake by the adrenal cortex in response to a hypothalamic stimulating factor, ACTH. All right? So now I am slowly depleting the LDL particle, and I'm doing the job I'm supposed to be doing. I'm delivering cholesterol to tissues. There is now this is where genetics, environment, and dietary style, lifestyle makes a difference, because it turns out that depending on not only the concentration of LDL, but the possibility, and I'm just going to draw a little asterisk here, suggesting oxidation. Let's just argue that the longer LDL particle stays in blood, the more likely it can get oxidized by redox potential, anything, reactive oxygen species. And this can include cholesterol, forming oxysterols, or even fatty acids on any of the lipids. This is what we're talking about in terms of pathology. Because it's a combination of the amount of LDL, how long it stays in the blood, and how prone it is to undergo some sort of oxidation, because this is recognized by the body's immune system as being naughty, bad. So I have to get rid of it somehow. All right. Normally, in a healthy individual who's a reasonable diet and exercise enough, this is pretty much cleared by the liver. So this, again, this is eventually clears the LDL particle completely. In the interim, though, is when things could happen. So now I'm going to just try and point out where possible damage could happen. All right, so we talked about the tag-rich particles. We can argue either for an increase in tag as a result of, let's say, lipoprotein lipase mutation, lipoprotein lipase suppression, ApoC2 mutation or suppression, ApoC3 increases. So all of these, you can envision how you would see accumulation of tag in blood. Hypertriacylglycerolemia. Hypercholesterolemia, most of the time, is attributed to this fellow. And what are the causes, again, of accumulating LDL cholesterol? Very similar. So the most common are LDL receptor disorders. All right? And you can argue many things. You can say LDL receptor number is reduced. Let's say mutation in the sequence. All right? So quantity, quality. You can also argue that there's something wrong with, remember ApoB100 is here. And half of it is the binding domain. So you can argue LDL ApoB100. All right? And finally, the fourth reason could be ApoE. Remember I told you that binding to this receptor is, let's say 70–80% binding through the LDL receptor through the ApoB100, but ApoE, and I'll just do it here, contributes to that binding. So a final factor in the possibility of causing hypercholesterolemia would be something wrong, either a deficiency or a mutation in ApoE. And this will come back when I talk to you about the causes of hypercholesterolemia. And there are 4 possible reasons why LDL receptor could be defective. Let me just slide this for you. Okay. Can you see guy in the back? Can you see this? No nods. Anyway. All right, so again, this would accumulate if you have something wrong with the pathway of uptake of LDL into the recipient cells. So anything can interrupt this. There's also, don't forget, a possibility of making more LDL than it should be. So any increase in, let's say, in VLDL would lead to an accumulation of LDL. So that's sort of a concentration factor. What else? There is a possibility, again, of something wrong, let's say, with the liver's clearance, finally, of the LDL. Okay? So we've spoken again about the sort of dichotomy of chylomicron fate versus VLDL fate. One thing we haven't spent some time on is the maestro of it all, and that's the HDL, or high-density lipoprotein component. All right, so again, most of HDL comes from liver, but a small amount comes from the small intestine. The major structural protein is that large dimer called ApoA1. This is the deflated looking tire that's secreted, and the HDL particle looks something like this. So here are the 2 ApoA subunits. And of course, all those other ApoCs and ApoE that's associated with these particles. But from a structural standpoint, this is critical, because this is really not only the major structural component of HDL, but it also has catalytic functions. It again speaks to the tissues. So briefly, what happens to an HDL particle is born literally, and then let's just call it HDL3, 3 being the least mature form. And when it acquires cholesterol from tissues, this is, I'm going to just say normal metabolism. Cholesterol, remember, is amphipathic. So cholesterol sits on the outside, and there's an enzyme associated with HDL3, with all forms of HDL, called LCAT, that will take cholesterol and make cholesterol ester. And this taking involves a fatty acid from phosphatidylcholine, also called lecithin. Hence the name lecithin cholesterol acyl transferase. It's a small amount, so the lysolecithin that's formed isn't really harmful. But now you've created a very hydrophobic particle as opposed to its amphipathic precursor. So cholesterol moves from the outside to the inside and creates now, slowly, will fill up the HDL particle with cholesterol. So whatever cholesterol is on the outside, eventually most of it will end up like this. And that's the mechanism. It's like a cargo ship filling up with, and the whole process is simply the activation of LCAT by ApoA, so that it will acylate cholesterol into cholesterol ester and fill up the hole. Now, there's one point, because if you don't do this, this for some reason can be cleared by the kidney. In other words, it does not, it is, most of the stuff that goes through the kidney is secreted and reabsorbed in the distal glomerulus. This is one of the few things, or one of the things that don't get reabsorbed. So you would lose a lot of the HDL particle if it didn't undergo this maturation. All right? So again, this is a critical part of keeping HDL in circulation by these facets. ApoA1 telling tissues, give me cholesterol that you don't need. ApoA1 stimulating LCAT to esterify that cholesterol, and this particle now can eventually go to liver. And liver has a specific called scavenger SRB1 scavenger-like receptor B1. This is the receptor that binds and clears HDL particles. And that's why it's called reverse cholesterol transport, because it's taking cholesterol from tissues back to the liver. Okay? All right, so just in case now, I'll throw in a little bit of pathology. The final component is the role of ApoA1 to, let's say, repair if this cholesterol or any other lipid it's acquiring happens to be oxidized. Right? So again, this is recent studies have shown that the ApoA1 can literally reduce oxidized sterol into, let's say, good cholesterol. Because in circulation, before this is cleared, there's another enzyme that will take, so let's again just draw the HDL particle. There's an enzyme that will actually exchange, it's called cholesterol ester transfer protein. It'll interact with any of the TAG-containing precursors. So VLDL, LDL, even IDL for that matter. And it will take TAG. And the argument is, it's going to take, let's just argue that it takes oxidized TAG into the particle and swaps it now with good cholesterol ester. So let's say it originally got oxidized cholesterol, ApoA1 repaired it, now it's shipping back. This, by the way, is one of the major causes of hyperlipidemia. For some reason, right? When you look at the demographics of hyperlipidemia, especially hypercholesterolemia, defects in this step seem to play a very important role, even though it doesn't seem to be a very critical step. But if you argue that, again, part of the pathogenesis of hyperlipidemia is this becoming a plaque, you can see how a repair mechanism would obviate that, would get rid of it. All right, so again, if I'm arguing that ApoA1 does play a protective role in reducing